So let's think about this for a second. What if we could fight cancer, but not with the usual strategy? Not by carpet bombing the entire body with harsh chemicals. What if instead we could send in a microscopic army to attack it from the inside out? Now, I know it sounds like something straight out of science fiction, right? But it's not. This is the absolute cutting edge of a field called synthetic biology, and believe me, it's completely changing the way we think about medicine.
Think about it. For decades, chemotherapy has been one of our main weapons against cancer. And it can be powerful, for sure, but it's a blunt instrument. It's like swinging a sledgehammer. It goes after rapidly dividing cells, which sounds great, but it hits all of them, all over the body. And that's where those devastating side effects come from. But this new approach, what people are starting to call a living medicine, it's a whole different ballgame. It's not a sledgehammer, it's a surgical strike. It's a precision attack launched from inside the enemy's own fortress.
So, to really get why this is such a massive game changer, we have to talk about the problem we're up against. And honestly, the problem is that solid tumors are basically tiny, impenetrable fortresses. You see, a solid tumor, we're talking things like glioblastoma or pancreatic cancer, it's not just a simple clump of bad cells. Oh no, it builds its own defenses. It creates this dense, chaotic mess with a really poor blood supply. And that's a huge issue, because it means the drugs we pump into the bloodstream have an incredibly hard time reaching the tumor's core, the very place they need to be. It's this hostile little bubble they create that makes so many of these cancers just stubbornly resistant to treatment. All right, so if our drugs can't get in from the outside, how in the world do we attack the tumor? Well, the answer is kind of crazy. It comes from one of the simplest forms of life on the planet, bacteria. Yeah, bacteria. There are surprising little undercover agents in this fight. And this is where we find the tumor's secret weakness, its Achilles heel. It's a concept called hypoxia. Basically, the very core of a tumor is a low oxygen environment. For our healthy cells, that's a total dead zone. But for certain kinds of bacteria, these so-called anaerobic bacteria, it's paradise. It's the perfect vacation spot. They absolutely thrive there. And that's the genius of this idea. We're turning the tumor's greatest defense into its biggest vulnerability. Okay, so let's follow the journey of one of these little agents. Scientists take a safe probiotic strain of bacteria, something like E. coli Nissl 1917, and introduce it into the body. And what happens next is amazing. These things act like tiny self-guided missiles. They just cruise through the system, completely ignoring all the healthy, oxygen-rich tissues. But the second they stumble upon that low-oxygen, hypoxic core of a tumor, bam, they hit the brakes. They've found their new home. And they start to multiply like crazy, by the millions. They literally find the target all by themselves. But, just having a bunch of bacteria hanging out in a tumor isn't going to cure cancer. That's just step one. This is where the really cool part comes in, the synthetic biology. Scientists aren't just sending them in, they're programming them. They're turning them into tiny on-site drug factories. How do they do that? Well, using incredible tools like CRISPR, they literally insert a new genetic circuit right into the bacteria's DNA. Think of it like uploading a new software program. This circuit is a new blueprint, a whole new set of instructions. So suddenly, these bacteria aren't just harmless passengers anymore. Nope, they've been given a mission. And that mission can be super specific. I mean, check this out. In one absolutely groundbreaking study, scientists programmed E. coli to manufacture an FDA-approved drug called Romidepsin. Now, this drug is what's known as a histone deacetylase inhibitor. That's a mouthful, but what it does is it basically flips a switch inside the cancer cells, reawakening their own self-destruct programs. It tells them, hey, your time's up. And the bacteria are building this potent weapon right there inside the tumor. Okay, I know what you're thinking because I was thinking it too. Is this safe, having drug-producing bacteria inside you? And the answer is a resounding yes. In fact, that's probably the most elegant part of this whole design. They build in multiple safety features. For example, the drug factory only switches on when it detects the tumor's unique low-oxygen environment. They also use this really clever trick called quorum sensing. It's like a biological head count. The bacteria literally wait until there's enough of them, a quorum, before they all start producing the drug at once. This ensures a powerful, coordinated attack. And get this, they can even be programmed with kill switches to self-destruct after the mission is complete. It's just brilliant. So, okay, the theory is brilliant, the design is elegant, but the big question is, does it actually work? Let's take a look at the results from the studies and see how this inside job actually played out. Okay, look at this number, 1.5 micrograms per gram. That might not sound like a lot, but in the world of pharmacology, it's staggering. In mouse models, this is the concentration of the drug that the engineered bacteria managed to produce inside the tumor. That is a seriously powerful therapeutic dose. And the key part? It was generated exactly where it needs to be, and almost nowhere else in the body. And the results, well, they're potentially life-changing. Let's talk about glioblastoma, one of the absolute deadliest brain cancers. In another study, a similar bacterial therapy increased the median survival time in the animal models by 45%. Let me say that again, 45%. That is not a minor improvement. That is a gigantic leap forward. And you can see it right here. This graph really shows you how powerful this is. That orange line, that's what you expect. Aggressive, unchecked tumor growth. But look at the blue line. That's the group that got the drug-producing bacteria. The tumor growth is just dramatically slowed down. The proof, as they say, is right there in the data. And here's the crucial takeaway, the thing you really need to remember. It's not just that it's more effective, it's that it's so much safer. This quote from the research paper says it all. Because the drug is only being made locally, inside the tumor, this whole method drastically cuts down on the awful side effects, like heart toxicity, that you get when you just flood the whole body with chemotherapy. I mean, this is the dream of precision medicine, made real. Okay, so this is an incredibly promising new frontier. But we're not quite at the finish line yet. So let's talk about the challenges that are still ahead and what the future could look like. You know, the path from a cool idea in the lab to an actual treatment in a clinic is always a long one. There are hurdles. Researchers have to be 100% sure that the genetic programming they put into these bacteria is stable for the long haul. Then there's manufacturing. How do you make enough of these living medicines for thousands of patients? And, of course, because this is a totally new kind of therapy, it's got to clear a really high regulatory bar, with agencies like the FDA before it can even get to human trials. But the potential payoff? It's just immense. Think about some of the deadliest, most difficult to treat cancers out there. Pancreatic cancer, glioblastoma. These are diseases where traditional treatments have really struggled. But this approach, it offers a brand new and incredibly powerful reason to be hopeful. It's a new way to finally breach that fortress. And that brings us to a final, pretty big thought. For centuries, right, we've looked outside our bodies for cures. We've used plants, chemicals, radiation. But what if the most powerful tools have been right here inside us, or all around us the whole time? By learning to harness and reprogram life itself, we might just be on the verge of creating the ultimate personalized weapon against our oldest and most challenging enemies.